So hi everybody, and welcome to your second lecture of the semester. Before we even start this lecture, I just want to level with all of you and be honest with you, this is going to be a long lecture. Every other week you have two separate lectures to watch for class, and this is the only lecture for this week because this is really like two separate lectures in one. I would say this is about a lecture and a half or a lecture and 3/4. So you are absolutely insane if you try to watch all of this in one sitting. I highly advise you to make the max amount of time you sit through one reading of this lecture, an hour and 20 minutes, just like you would in class. Try to treat this like a class in that sense, and then give your brain a rest and come back and finish the rest of it. I'll try to get a break worked in there at about an hour and 20 minutes and tell you this is a good place to pause the lecture and come back to it later. But again, this is almost like two lectures in one. This is all of Chapter 2. Chapter 2 is called Chromosomes and Cellular Reproduction in your textbook. We'll begin the lecture by just much more structured way delineating and identifying the differences between prokaryotic cells and eukaryotic cells, especially as they pertain to genetics. We'll talk about the basic requirements that a eukaryotic cell has for cellular reproduction, and by cellular reproduction we just mean cell division, one mother cell splitting into two daughter cells. We'll talk about mitosis and meiosis in some great detail, and I'd like to draw your attention right now to Khan Academy tutorials that are linked through the Canvas account, the Canvas website. So if you go to our course website in Canvas and then click on modules, one of the modules will be called Online Resources, and under that module you'll have links to a mitosis tutorial at Khan Academy and a meiosis tutorial. I recommend you use those if you find them beneficial. We'll end the lecture talking about the implications of meiosis on sexual reproduction and be ready for the rest of Mendelian genetics. So I've already given you the punchline here, but don't let the name of this chapter or this lecture fool you. We're talking about mitosis and meiosis today. We need to give quite a bit of background as to why mitosis is even necessary. So the first 20 or 30 minutes of the lecture will really be about that. Why is the process of mitosis even required in eukaryotic cells? Why is it not required in prokaryotic cells? But then we'll launch right into the nitty-gritty of these processes. When one cell divides into two, in any case, regardless of what form of life we're discussing, each new daughter cell must have an exact complement, an exact set of the contents of the one mother cell. When we have cellular reproduction, the daughter cells should be nearly identical to what the mother cell was. For the cytoplasm, this really isn't a problem. The cytoplasm can be divided in half. The mother cell says, you get some and you get some. Keep in mind, after cellular division, there is no mother cell anymore. Don't think of it as terms of the human mother, where the mother exists and the offspring are there. One cell splits into two smaller cells. The initial cell is the mother cell. The two smaller cells are the daughter cell. Once cellular division has occurred, there ain't no more mother cell. That mother cell has been split into two. So we can divvy up the cytoplasm by just dividing the cytoplasm in half, and each new daughter cell gets half of that cytoplasm. Same for the organelles. The mitochondria, the endoplasmic reticulum, same idea. You get some, and you get some. But see, there's a problem with the nucleus. The nucleus is the one organelle in the cell that is unsplittable. There is only one. So how do you take one nucleus and give one to one daughter cell and one to the other? The only way to give each new daughter cell a nucleus, exactly like the one the mother had, is to copy that nucleus, copy the DNA inside of it, turn one nucleus into two, and then give one each to the new daughter cells. Now, the nucleus is the home of the genome. The DNA exists in the cell's nucleus. So if we're copying the nucleus, what we really mean we're copying is the DNA that it contains. That is the real crux of mitosis. So why is mitosis needed in a eukaryotic cell and not a prokaryotic cell? Well, to have that discussion, let's first be sure that we're absolutely confident what the differences between these two cell types are. Mitosis is the name of the process that eukaryotic cells, and only eukaryotic cells, use to ensure that each new daughter cell gets its own nucleus with the exact same DNA content, the exact same chromosomes that were present in the mother cell. Meiosis is also a strictly eukaryotic process, but this is the name of the process that's used to obtain cells containing exactly half the number of chromosomes that were in the mother cell. And by half, we mean one of each chromosome pair. That'll become a little bit clearer as we get later on in the lecture. But meiosis splits the DNA content of a cell in half. What we have as a result of meiosis is a sex cell, or more appropriately referred to as a gamete. The gamete contains a nucleus that contains half the DNA of the mother cell. And this is important for sexual reproduction. So this raises an important point already. Prokaryotic cells replicate, they divide, they undergo cellular division, but they don't do mitosis or meiosis. Why? Well, one reason is that prokaryotic cells don't contain a nucleus. So we've already said that one of the main points of mitosis is that eukaryotic cells have only one nucleus. And if you want to give one nucleus to two daughter cells, you've got yourself a problem. Prokaryotic cells don't have a nucleus. And so they don't have this confine of having to turn one nucleus into two. In addition, prokaryotic cells only have one, usually circular chromosome. So once you copy that single chromosome into two through DNA replication, you just give one of those new chromosomes to one daughter cell and the other to another. That makes DNA replication fairly easy. Eukaryotic cells usually contain many, many linear chromosomes. Each linear chromosome must be replicated, and each is unique, different from the others, and each is essential. In other words, if a daughter cell doesn't get exactly one of each copied chromosome, it's in trouble. This is a much larger problem. Here's the analogy. I don't know how many of you play Uno. Uno's a big thing in my house. My younger daughter not only loves it, she's a card shark. She's 5, and she kicks everyone's butt in Uno. But the way the Uno deck works is there are actually two identical Uno decks, and you shuffle them together to give one big deck. That means there's 2 red 3s, 2 green 3s, there's 2 of everything, okay? So imagine this is how it works in a eukaryotic cell. You have a single Uno deck. That's the genome content of the mother cell. Now you undergo DNA replication. So now you have 2 of every card, 2 of every chromosome, and they're identical. That's your second Uno deck. That's your second green 3, your second red 4. Now you've got 2 of every. Okay? But it's a mess in the nucleus. So now imagine those 2 Uno decks are very well shuffled together. So they're all mixed up in the nucleus, okay? The problem comes when it's time to separate those decks again for cell division. What you need to do in mitosis and cell division is ensure that one daughter cell gets exactly 1 red 3, 1 red 4, 1 red 5, 1 green 3, 1 green 4, 1 green 5. In other words, make sure that one daughter cell gets exactly 1 full Uno deck, and the other daughter cell gets the other full and complete Uno deck. Can you imagine the logistical nightmare that is? To first find the 2 green 3s, pair them up, and then make sure that 1 green 3 goes to one new cell and the other green 3 goes to the other. That's a mess. In bacterial cells and prokaryotic cells, you have a deck of 1 card. Here's your green 3. You undergo DNA replication, you have 2 green 3s. You're going to undergo cell division, one cell gets 1 green 3, the other cell gets the other. Done. You don't have this mess of a shuffled deck of identical cards that needs to be faithfully and accurately dealt out to 2 daughter cells. So that is the problem between the 2 cell types. There are other differences that you're aware of, I'm sure. Prokaryotes are much smaller, eukaryotes are larger, prokaryotes don't have organelles, eukaryotes do, etc., etc. But for us, the real point comes down to that nucleus and the number of chromosomes. Eukaryotic cells have a nucleus and prokaryotic cells don't. Eukaryotic cells have many, many linear chromosomes. Prokaryotes have only 1 circular chromosome, a single card in their deck. But eukaryotes and prokaryotes do have one major thing in common, and that is that both cell types, bacteria and eukaryotic cells, have more DNA in their genome than can fit in their cell. Now, wait a minute. What did I just say? Yeah, it's true. Bacterial cells and eukaryotic cells have much more DNA than can physically fit in their cell. There's 6 feet of DNA in each one of our cells, okay? And obviously each one of our cells is much smaller than 6 feet in diameter. So how do we work around that? Well, it's actually a pretty simple fix. Think of my office. If you haven't been in my office yet, I encourage you to stop by. We'll have a nice conversation, but I'm going to guess that my office is about 20 feet long and 15 feet wide, roughly. I'm just guessing. That's a typical office size, I think. So you could say that if I had 100 feet of rope, 100 feet of rope doesn't fit in my office. My office is only 20 feet long and 15 feet wide, and 100 feet of rope doesn't fit in my office. And that's the same as saying 6 feet of DNA can't fit in a single cell. But of course you're going to say, what are you, ridiculous? Of course you can fit 100 feet of rope in your cell, and just wrap it up. Make it compact. Make it dense. Wrap it around your arm, wrap it around a spool, wrap it around something, cause that rope to not be 100 feet all stretched out, but be a much smaller size all compacted in, and then you can easily fit it in your office. And you would be exactly right, and I would feel like a fool. So this is exactly the approach that prokaryotic cells and eukaryotic cells take. They wrap their DNA up. It's just that they do it in very different ways. Each cell type wraps their genome up, compacts that DNA to get it to fit in the relatively smaller size of their actual cells. It's their approach that differs. Eukaryotic cells wrap their DNA around protein spools, true spools made of proteins. The proteins are called histone proteins. And these histone proteins come together to form a cylindrical spool, and the DNA wraps around that spool, just like string around a sewing spool, creating structures that are referred to as nucleosomes. So just as we would wrap this twine around a cardboard spool, we wrap our DNA around a protein spool, giving nucleosomes. In fact, this is what's shown on the cover of the textbook we use for this course. The DNA is in blue, and the histones are in orange, and together they wrap together, forming nucleosomes. And that's how the DNA is compacted. Individual nucleosomes are compacted further to eventually create chromosomes. We go through some of the details of this in cell and molecular biology. It's not important for genetics, but this allows us to get from 6 feet of linear, unwrapped, uncompacted, what we refer to as naked DNA, wrapped around histone proteins, further compacted, further wrapped, further compacted, further wrapped, until finally we have all of that DNA wrapped so tightly into the chromosome structure that we're more used to seeing in our textbooks, this structure that we see down here. Eukaryotic DNA that's packaged in this way in nucleosomes is referred to as chromatin. Chromatin is packaged DNA. And actually, I'm going to get out of the lecture for just a quick second, but some of you may wonder already on my screencast here what this is on my wallpaper, what this is in the center of my computer, and this is a nucleosome. This is the proteins here in the center in blue, yellow, light blue, and gray. These are histone proteins, and the DNA is wrapped around the outside of that protein spool. I've done quite a lot of research over my career on nucleosomes, and so they are near and dear to my heart. Prokaryotes, bacterial cells, don't do this. They compact their DNA, but they don't use nucleosomes. Prokaryotic cells have circular genomes, and so they have a much easier time compacting their genomes. All they have to do is a process called supercoiling. Now I'm going to ask you to pause the lecture and go get yourself a rubber band if you can. Any rubber band will do. I'll wait here patiently while you do that. Now that you're back, I'll assume that you found the rubber band, and I'm going to talk you through some supercoiling. What you're about to do is exactly what bacterial cells do to compact their DNA. If you are right-handed, I want you to grab one end of the rubber band with your left hand between your thumb and pointer finger and squeeze that rubber band tightly. Don't let go. If you're left-handed, just hold the rubber band in your right hand. Don't let go. Now with your other hand, the hand that's not holding the rubber band, I want you to lightly grab the other end of the rubber band and twist it. And without kind of letting go of the tension you're creating, keep twisting it. Twist it and keep twisting it, twist it and keep twisting it. And you're going to find that that rubber band begins to kind of knot up and coil up in on itself. And in no time at all, it takes up much less volume, much less space than it was taking up before. Now you can let go of that rubber band and it might unravel all on its own, or you can help it along and get it back to an unbroken circle of rubber. But you can repeat the process, squeeze it between two fingers, and with the other hand, curl and roll, curl and roll, curl and roll, and get that rubber band all super coiled up. And you have just demonstrated for yourself how bacterial cells package their DNA. It's exactly what they do. Exactly what they do. I hesitate to even call this an analogy because it's exactly what they do. This only works for circular structures. You try cut the rubber band in one spot, make it linear, and try twisting it, nothing's going to happen. You could be twisting all day. And so our DNA, our linear chromosomes, need to be wrapped around nucleosomes. So let's review a few key points that we've already covered here before we move on. Make sure that everybody's okay and comfortable. First, we have linear chromosomes, while prokaryotes have single circular chromosomes. We have nuclei, while prokaryotes do not. The implications of these observations is that cell division is a much simpler process in bacterial cells. We actually don't call it cell division, we call it binary fission, but that's okay. No nucleus means that you can just take your single genome, copy it, and then deal out that deck of 2 cards saying, you get a chromosome, and you get a chromosome, and we're done. So for a bacterial cell, you take that one single chromosome, you divide it or copy it into 2 chromosomes. One of those chromosomes sticks to one end of the mother cell, the other chromosome sticks to the other end, and as the mother cell divides into 2, the 2 chromosomes are divvied between them. Easy peasy, bumpin' easy. It's so easy and straightforward that a bacterial cell can actually divide every 20 minutes under optimal conditions because it doesn't need to spend a ton of time dealing out a large deck of copied chromosomes. Our cell division is much more complicated in eukaryotic cells. First of all, we've got this nucleus that we have to get rid of before we can even begin to reproduce cellularly, so we got to dissolve the nucleus. Each chromosome needs to be replicated. That's each chromosome needs to be replicated. We have 46 chromosomes in our cell. Each one needs to be copied. Then we've got to deal out these new copied chromosomes so that each daughter cell gets one copy and only one copy of these newly replicated chromosomes. That's the separating of the UNO decks, making sure that one cell gets one green 3. And now we got to look through the deck. Where's that other green 3? Where's that other green 3? Okay, the other cell gets the other green 3. Now on to the 4s. One cell gets one green 4. Where's the other one? Where's the other one? Oh, here's the identical top copy. The other cell gets the other green 4. What a pain. Once that's all done, you got to make new nuclei. You got to reform the nuclei for each of the new daughter cells, and then the cells actually have to split and divide, giving rise to 2 independent daughter cells. It's a mess. It's a mess. DNA replication itself is a complicated endeavor. It's complicated in both cell types, prokaryotes and eukaryotes, but that's not for this course. Genetics doesn't focus on DNA replication. You'll learn all about DNA replication in cell and molecular biology. All right, so on to eukaryotic chromosomes. We know a little bit about them. They're linear. They need to be wrapped around histones to create chromatin. There's a lot of them, so we need to deal them out. Different eukaryotic species have different numbers of chromosomes, and more chromosomes, higher chromosome number, by no means correlates to greater complexity of the organism. We've already said a number of times, and I'm sure you've heard it in other courses, that we, human beings, have 46 chromosomes, 23 from mom, 23 from dad. The small diminutive house mouse has 40 chromosomes, 20 from mom and 20 from dad. Fruit fly has 8 in its diploid state, 4 from mom and 4 from dad. Your dog, which may be superior to you in many ways, but trust me when I say it's not superior to you in the ways that matter, has almost twice as many chromosomes as you do, 78 chromosomes for the domesticated dog. Same number for the chicken, which I don't know about you, but I'm better than every single chicken I've ever met. The crayfish, the large shrimp-like thing that lots of people like to eat, has 200 chromosomes. And a small plant called the field horsetail plant, 216 chromosomes. So never should you believe that the number of chromosomes correlates to the complexity of the organism. The ant has 2 chromosomes. The muntjac, which is a kind of a deer-like thing, has 6 chromosomes. We have 46, and the crayfish has 200. Okay, but we have 46 chromosomes. That much we know for sure. And most eukaryotes have 2 sets of chromosomes. This is why the numbers that you see in front of you are even numbers. Those 2 sets come because one set came from your mother and one set came from your father. The lowly ant got 1 chromosome from its mommy ant and 1 chromosome from its daddy ant, but that gave baby ant 2 chromosomes. We get 23 chromosomes from mom and 23 from dad, giving us our full set of 46. So most eukaryotes do have an even number of chromosomes because they're doubling the number of chromosomes that they get from each parent. Each parent contributes half of their own genome. We call this state the diploid state. Diploid, for two of. What we have two of here is two sets of chromosomes, one from each parent. This brings us back to the idea of alleles. So your mother gave you a chromosome number 1, and your dad also gave you a chromosome number 1, giving you a chromosome pair for the first chromosome, one from mom and one from dad. The chromosomes that your parents gave you for chromosome 1 are very, very similar, but they're not necessarily identical. Each of them contains the same genes in the same place. Remember what genes are. Genes are things such as hair color, eye color, the quality of your vision, etc. But they could, by they I mean the chromosomes that your mother and father gave you, they could have different alleles, different versions of those genes. You remember as well as I do, Wide Awake can come on vinyl, cassette tape, CD, or MP3. It is the same information in a different version. Hair color can come as red hair, blonde hair, brown hair, black hair. It's the same gene, hair color, but different versions of that. And so you have 23 pairs of chromosome. You got this chromosome number 1 from your mother and this one from your father. You got this chromosome number 2 from your mother and this one from your father. Mom gave you this and dad gave you that. Mom gave you this and dad gave you that. Mom gave you one of each of these 23 chromosomes and dad gave you another set, giving you your 46. But if we zoom in on chromosome number 1, this one came from mom and this one came from dad. Here is one allele from your mother, and here is another different allele from your father. Same information, but different versions. Mom gave you brown hair, and dad gave you blonde. Same information, hair color, different version. Brown hair, blonde hair. Now this raises a very big question. If mom gives you brown hair and dad gives you blonde hair and you can't have both at the same time, how do you know which one wins? How do you know what type of hair you have? And that is Mendelian genetics. The answer to that question is all that Mendel contributed to the field of genetics, and it's where we'll go after the material in this lecture. That is the crux of genetics, the winning of one allele over another. And we'll go over that in some detail as we get there. To back up a second, though, in some terminology, we call these two chromosomes, the two chromosomes number 1, a homologous pair. Homologous meaning similar but not necessarily identical. Same information but different versions. So you have a homologous pair for chromosome number 1. You also have a homologous pair for chromosome number 2, for chromosome number 3, for chromosome number 4. You have homologous pairs for all 23 chromosomes. I do not. I have a homologous pair for the first 22 chromosomes, but I am male. My 23rd chromosome pair is actually one X and one Y that are not similar at all, therefore non-homologous. We'll come back to that in just a second. But this term homologous pair means that the two chromosomes that you get, one from mom and one from dad, are extremely similar but not necessarily identical. So let's come full circle. Let's go back to these ideas of meiosis and mitosis. The process of meiosis takes a cell, and to kind of boil it down almost to the point of inaccuracy, takes a cell in a human being and has that cell divide without first replicating the DNA. We all normally have 46 chromosomes in each of our cells. If a cell divides without replicating its DNA, how many chromosomes would it have? Well, you would take 46 chromosomes and split it in half. Half goes to one cell, half goes to the other. That's 23. So now you have a cell with 23 chromosomes. Now, if I take one of my 23 chromosome cells and I fuse it with one of my wife's 23 chromosome cells, how many chromosomes does that new cell have? Well, 23 from me, 23 from my wife, 46 total. There is the new human being. We've reconstituted the 46 chromosome number. I gave one chromosome number 1, one chromosome number 2, one chromosome number 3, one chromosome number 4 to this new life. My wife gave one chromosome number 1, one chromosome number 2, one chromosome number 3 as well. Those two sets came together to create 23 homologous pairs, And I can say that because I have 2 daughters, so 23 homologous pairs, including the XX 23rd pair, and that was the new human being. We just described sexual reproduction as boringly as humanly possible. In fact, not to digress, but this is exactly how I described sexual reproduction to my 6-year-old daughter a year ago when she asked where babies came from. And it bored her so much that I doubt she'll ask again anytime soon, and that was the whole point. In any event, we have a human cell, a haploid cell, meaning it contains half, haploid for half the amount of genetic information that a typical human cell needs. One male human haploid cell fuses with a female human haploid cell, giving rise to a human diploid. Again, diploid for 2, a diploid cell that contains 2 of every chromosome, the 46 pairs. So sex cells, also known as gametes, or eggs for females and sperm for males, are made through meiosis to split that chromosome number in half to give rise to haploid cells, and then those haploid cells can come together and fuse to create a diploid human being. All right, so that's all that background. Now we're going to change gears a little bit and start setting the stage for mitosis itself. We'll give a little bit more background on the events that lead up to mitosis, but I hope that you're all comfortable with where we're going and why we're going there, and how many chromosomes we need, and how those chromosomes have to behave, and all of that good stuff. So most of the time in a normal living cell, chromosomes are very lightly compacted. The chromatin is not very dense. The chromatin fibers are quite thin, and they're nearly invisible under a normal light microscope. However, just before mitosis and just after DNA replication, chromosomes begin to compact very tightly. That chromatin gets very, very dense, and so dense, in fact, that the chromosomes become actually visible under a typical light microscope. This is what we're used to seeing. This is a real chromosome taken with an electron micrograph, just so we can see some detail. All these little fuzzies coming off of this chromosome are not as efficiently wrapped DNA, but this is just super dense, tightly coiled DNA, wrapped up very tightly. And this is the more kind of typical schematic view that we're used to seeing in textbooks of a chromosome as well. It's all just very densely wrapped DNA. A chromosome in this state has 3 main parts. It has a centromere, which we're going to talk about quite extensively in just a moment. The centromere is where the cellular machinery attaches to the chromosomes, allowing those chromosomes to be pushed and pulled during mitosis, allowing the cards to be dealt out. If there's no centromere, then the cell has no way to grab on to a chromosome. Without a way of grabbing onto the chromosome, those chromosomes can't be dealt out or separated, and that leads to really big problems. Another part of the chromosome, which we won't talk much about in this course, but you should see it because it'll be labeled in your textbook, are the telomeres. The telomeres are the ends of chromosomes. They protect the chromosomes from becoming chewed up and degraded on the ends. They're caps on the chromosomes. You can almost think of these as, I think these are called aglets, the little plastic things on the end of your shoelaces. I don't know why I know that name, and I could be wrong, but this is a good analogy for a telomere. With the cap, the end of the fiber is protected. The DNA doesn't degrade. It doesn't fray. Without the telomere, we do have DNA damage at the ends of our linear chromosomes because that protection is lost. Finally, there's an origin of replication on each chromosome, actually many on each chromosome, and this is where DNA replication is begun. But we won't talk about origins beyond this one reference because that's much more of a molecular biology concept. So centromeres are what we're going to be most concerned with in this lecture. So here we are. Here's our schematic. You can see that everything here is labeled. We have the telomeres on the ends, one telomere on one end, another telomere on the other. We have our centromeres roughly in the middle, although they're a little bit off-center sometimes. This is a single chromosome right here that we see. Once DNA replication has occurred, we have 2 identical copies of that chromosome. Now hold on, because this is going to get slightly confusing, and it's important you get this down now rather than struggle and suffer with it later. This is one chromosome. Actually, let me do some writing. This is one chromosome prior to DNA replication. This is one chromosome after DNA replication. The terminology does not change. A single independent molecule is a chromosome, However, this is a single sister chromatid, and this is another sister chromatid. Okay, so one chromosome is replicated to become two attached sister chromatids, which is still referred to as one chromosome. Alrighty? Okay. Okay. So after replication, but prior to identical copied sister chromatids being pulled apart, those copied DNA molecules are referred to as sister chromatids, and the single molecule of them stuck together is still called a chromosome. Once the two sister chromatids are separated and pulled apart, they are then two separate chromosomes. So it's all about whether they're attached or not. So what are we looking at right here? What we're looking at right here are 46 chromosomes. Okay? Unreplicated. 23 pairs of chromosomes. One from mom, the other from dad. One from mom, the other from dad. These are homologous chromosomes. After replication, we are looking at 46 chromosomes. Still 46 chromosomes. Each chromosome, however, now consists of two sister chromatids. These two chromosomes, this one and this one, are homologous, not identical, from mom, from dad. But the two molecules that are stuck together, the two sister chromatids, are the result of DNA replication, and they are identical. This is one green 3 of an UNO deck. This is the other green 3 of the UNO deck. This is a red 3 that you got from your mother, and this is the other red 3. Are they similar? They are. They're both 3s. Are they identical? No. One came from your father and is green, and the other came from your mother and is red. Okay, so that's what we mean when we say replication lays the groundwork for sister chromatids. Let's get started. The cell cycle, which includes mitosis, the cell cycle is essentially the life of a single cell. It contains multiple stages, some of which are the process of mitosis and cell division. Dropping all of the terminology and all of the complexity of the cell cycle really boils down to these simple steps. G0 phase. G0 phase is this little kind of detour out of the cell cycle. G0 is when the cell does all of its cell stuff. This is the non-dividing phase. You have a lung cell. Most of that time, that lung cell is gathering oxygen from blood, shuttling that oxygen to the rest of the body through the bloodstream, taking up carbon dioxide from the incoming blood, and throwing that carbon dioxide out so it can be exhaled. That's the job of a lung cell, and the lung cell does all that stuff in G0. However, when it's time for the lung cell to divide, it must fall out of G0 and enter the cell cycle process proper. Other cell types do other things in G0. Immune cells fight infections, neurons transmit electrical impulses, etc., etc. The rest of the cell cycle is truly for cell division. In G1, the first true step of the cell cycle, cellular preparations are made for copying the genome for DNA replication. G1 leads directly into S phase. S phase stands for the synthesis phase, and what you're synthesizing is a new genome. So in the S phase, DNA replication occurs. Now that all of the decks in that UNO deck, all the cards in that UNO deck have been copied, you now must prepare yourself for the dealing out of the two new decks. So G2 is used for preparations for mitosis, the actual separating of these two copied genomes. And then the M phase stands for mitosis. M for mitosis, that's where mitosis occurs and cellular division occurs as well. And then we've come around the full cycle. If the cell needs to divide again very rapidly, it's going to launch right back into G1. Prep for another round of DNA replication, do that DNA replication, prep for another round of mitosis, do that mitosis. If it was just a single division and now the 2 new lung cells just have to be lung cells, after mitosis and cell division, the cell will go back into G0 and happily be a lung cell. Either way, no matter how you cut it, after M phase, you have 2 new cells, and those 2 new cells can repeat this process as needed. Hence, the cell cycle. Cycles go on and on. They constantly repeat. And this is the cell cycle for repetition. Now, it shouldn't surprise you to know that lots can go wrong in the life of a cell. Things happen. We have DNA damage. We get infected with a virus. We have a loss of ATP production and the cell is starved for energy. Lots of things can go wrong. And if any of these things do go wrong, that's a bad cell. That cell's got some problems. And the last thing that cell should be doing is replicating. If it's got DNA damage, we don't want to transmit those mutations to any further cells. If it's got a viral infection, hey buddy, you take care of that first and you come back to me when you're healthy again. A cell that's starving for energy, my God, one of the most expensive, energetically expensive things a cell can do is DNA replication and cell division. So if you're hurting for energy, don't you be doing any DNA replication. Get yourself some energy and then we'll talk again later. So cells that are experiencing any of these insults should not progress into the cell cycle. Therefore, there are things called checkpoints, truly referred to as checkpoints during the cell cycle, where the proteins, the infection state, the energy state of the cell is monitored. And it's verified from the cell that the cell is healthy. It's almost like a self-diagnosis that the cell does. Am I ready for another round of replication? Should I really be doing this? Should I be dividing? Only when the cell proceeds through the checkpoints is it kind of verified or stamped that it's ready to proceed through the cell cycle, that it's appropriate for the cell to proceed through the cell cycle. The cell cycle can be divided into 2 broad phases. One is interphase, where the cell is doing everything it needs to do except dividing. So, said most simply, interphase is G1, S, and G2. All of that is interphase. As you see here on this diagram, all of this is interphase. And then the rest of the cell cycle is mitosis and cytokinesis. Mitosis is the dealing out of the replicated chromosomes, and cytokinesis refers to actual cell splitting. So let's talk a little bit more about interphase. Interphase is often referred to as the first stage of mitosis. So we have officially begun the mitosis discussion in this lecture. Although we really haven't done anything mitosis-y yet, interphase is the first step because it includes DNA replication. The textbook says that interphase is the, quote, extended period of growth and development between cell divisions. And I reproduce that quote here because I couldn't have said it better myself. That's what interphase is. It's the gap between cell divisions where the cell does everything it needs to do in order to divide again. Interphase looks boring as sin under the microscope. It looks like the cell is just sitting there dormant. Mitosis is very dynamic, very exciting. Things are flying to all different sides of the cell. The cell is splitting, it's pinching, it's doing all this crazy stuff. Interphase looks boring. But actually interphase is much more dynamic. It's just that the things that are moving around, the things that are accomplishing cellular tasks during interphase are so small and at the molecular level that you can't see it under a light microscope. But truly the cell is living and doing its particular things in interphase. Again, by convention, the cell cycle starts with G1. Here the cell is growing, it's making the proteins and enzymes that it needs for DNA replication. G1 lasts about 10 hours. G1 lasts about 10 hours. Bacterial cells do the whole cell division, the whole thing, in about 20 minutes, and G1 alone takes 10 hours. After that, the cell must pass through its first checkpoint, the G1S checkpoint. It's at this checkpoint that the cell measures and makes sure, do we have everything we need for DNA replication? Should we copy our genome? If the checkpoint is passed, we enter S phase. Again, S for synthesis, and here the genomes are copied. In the human cell, this takes about 9 hours. 9 hours to copy 46 chromosomes. Insane. So we're up to 19 hours now. We haven't even begun dividing. It's in the S phase that we do this 1 chromosome to 2 sister chromatids duplication, giving us two identical copies of each of our chromosomes. Before S phase, we have one molecule, and after S phase, we have two identical molecules. Whether we refer to it as a single chromosome or not, the fact of the matter is we have two identical copies after S phase, each of them referred to as a sister chromatid. Once that's been successfully completed, we move on to G2. G2 is where we prepare for the actual cell division and mitosis. It takes about 4 hours to do those preparations. We're up to, 23 hours already. The cell must then proceed through its next checkpoint, which is the G2M checkpoint. Have we done everything we need to do for mitosis? Should we really initiate cell division? When that checkpoint is passed, we move on to M phase. Interphase is officially done, and cells are ready to move into mitosis. So let's do that ourselves. Let's move into mitosis. So before we actually start covering mitosis as a process, I do want to bring to your attention that I expect you all to know all of the stages of mitosis. You need to know the names of the stages and the major events that occur in them. Don't be too intimidated. You'll see as we go along that it actually makes some intuitive sense what has to happen first and second and third. And I'll give you a useful mnemonic in just a second that'll let you remember the order of the stages pretty easily, I hope. But still, you need to be prepared on any exam to be fluent with these stages, their order, and what happens in them. So where are we? What have we done already? Well, we've finished interphase. Part of interphase is S phase of the cell cycle, so that means we have replicated our genome, we've copied our chromosomes, and so we have sister chromatids, identical copies of those chromosome information of that DNA. We have sister chromatids joined together into single chromosomes. So we've replicated our DNA, but the two copies of each chromosome are stuck together as sister chromatids. These sister chromatids are truly physically stuck to one another, and what's holding them together is a protein called cohesin. Cohesin is like a band, a rubber band, around the two identical sister chromatids, keeping them together. If we have any hope at all of dealing these identical chromosomes out to either side of the mother cell, to the two new daughter cells, we need to somehow get rid of this cohesin and separate these sister chromatids into individual independent chromosomes. That's what we need to do in mitosis. And then we have to play this game of literally dealing out those identical copies. You get one of my dad's chromosome 1s, and you get the other identical copy. You get one of my mom's chromosome 1s, and you get the other identical copy. That gives you one homologous pair, and that gives you the other homologous pair. Dealing them out one by one so that each new daughter cell has the exact same genetic information. That's what mitosis is. It's the process of getting one sister chromatid to each new cell. And mitosis is divided into 5 stages. The M phase of the cell cycle is mitosis. All 5 stages of mitosis make up the M phase of the cell cycle. The 6th stage of the M phase is referred to as cytokinesis. Cytokinesis is cell splitting. That's what that word means. That's when the mother cell truly divides in half, creating 2 new daughter cells. Before we begin to discuss the stages of mitosis, I want to take just one quick step backwards and remind ourselves where we are. We're at interphase. Interphase is done. The DNA has been replicated. The nuclear envelope is still intact. The cells still have a nucleus. And the DNA has just started compacting into its very compact mitotic chromosome state. So our first true phase of mitosis is prophase. As cells leave interphase and enter prophase, the DNA fully compacts and for the first time becomes so compact that it's visible under a light microscope. Remember, when we say these chromosomes, we mean identical sister chromatids held together by cohesin. So we are looking at these 2 sister chromatids held together as single chromosomes. Also, a pair of centrosomes move to opposite sides of the cell. We're talking about chromosomes, we're talking about centrosomes. Try your best to keep the terminology straight. I know there'll be a challenge there, but do the best you can. What are centrosomes? Well, centrosomes contain proteins called microtubules. Microtubules are a type of protein cellular cable. What I like to think of centrosomes as are fishing poles, fishing lines, spools of fishing line. And these microtubules are the actual fishing line itself. The centrosomes is the fishing pole, the place that the fishing line comes from. From there, mitotic spindles form. What are mitotic spindles? Mitotic spindles are nothing more than the fishing lines going out and attaching to individual sister chromatids. So let's take a step back, and go back into your imagination, go back to your centrosome, which was the fishing pole, but now imagine that it is a magical fishing pole that can send out 46 different individual fishing lines. One huge monster fishing pole that can send out 46 fishing lines simultaneously. Each one of those fishing lines is a microtubule. Okay? All 46 of them out at once is the mitotic spindle. This huge array, this huge web, this huge set of many, many fishing lines, all coming out of one pole, all coming out of one centrosome, attaching to individual chromosomes. The textbook says the mitotic spindle is an organized array of microtubules. I translate to say essentially it's the cellular cables that will eventually attach to and pull the chromatids apart. But I think if you hold on to that mental image of a single large fishing pole as the centrosome, individual mitotic microtubules coming out of that, being each of the microtubule cables, those are your fishing lines, but all of those lines together making up this spindle, this mitotic spindle. You might also see referred to in the textbook and other resources that you might find for yourself, centrioles. I'm not going to talk about centrioles. It just overcomplicates the situation. For this purpose, you need to know what the centrosomes are, the microtubules, and how they relate to the mitotic spindle. Okay. So that's it for prophase. We've got the compaction of the DNA chromosomes. We've got the setup of the microtubules, the setup of the centrosomes, the setup of the spindle, microtubule spindles. Okay. Now we've moved on to prometaphase. The beginning of the prometaphase stage of mitosis is marked by the dissolving of the nuclear envelope. The nuclear membrane literally dissolves. It disintegrates. How does it dissolve? I'll tell you next semester in cell and molecular biology. For the purposes of genetics, just know that it dissolves. What was the nuclear envelope doing? Well, the centrosomes are in the cytoplasm. The microtubules, the mitotic spindles, formed in the cytoplasm. And so you cast out your fishing line prior to prometaphase, and the end of your fishing line hits the nuclear envelope. And you say, well, damn, I'm supposed to attach to a chromosome. Let me try that again. So I'm going to throw out my fishing line again, and I hit that nuclear envelope. It's like going fishing on the lake, and covering the whole lake is a tarp. You can throw that fishing line out as many times as you want. If there's a barrier between you and the fish, you're never going to get anything caught. If there's a barrier between the mitotic spindle and the chromosomes, that barrier being the nuclear envelope, you're never going to catch a chromosome. And so the nuclear envelope had to dissolve to allow the spindle microtubules to access and reach the chromatids. Typically, a microtubule from one centrosome will attach to the kinetochore of one chromatid, while a microtubule from the other centrosome on the opposite side of the cell will attach to the other sister chromatid of that chromosome pair. What the hell am I talking about? Okay, deep breath, back up, let's get the big picture back in our minds. We had a single chromosome. In S phase, we replicated it. That gave us a single chromosome with two sister chromatids. This was the centromere, the constriction of that single chromosome. As part of that centromere is a structure called the kinetochore. It's shown here in green. The kinetochore is a protein structure at the centromere. All the way out here, to the left of my computer screen and the left of yours, is a centrosome. And that centrosome sends out a single microtubule, part of the spindle microtubules, and that microtubule attaches to the kinetochore. On the other side of the screen, on the other side of the cell, all the way to your right, is the other centrosome, sending out another microtubule, which can now access this sister chromatid because we've dissolved the nuclear membrane, and it hooks its line, it hooks itself to the kinetochore attached to the other sister chromatid. So now we have this one chromosome attached or tethered on both ends by microtubules attached to centrosomes on either side of the cell. That's what we mean. And then an interesting thing happens. The microtubules coming from opposite opposing centrosomes begin to lengthen and shorten, playing a sort of tug-of-war game with each other, with the chromosome in the middle being the flag on the rope. So quite literally, this single chromosome with its two sister chromatids gets pulled towards the centrosome on the right, then gets pulled to the centrosome on the left, then gets pulled back to the right, then gets pulled back to the left. Each of these centrosomes, through their microtubules, pulling and tugging on that chromosome in the middle. Unlike a true tug-of-war game, though, eventually each microtubule is going to win, each centrosome is going to win, because the chromosome can and will split in half. Each sister chromatid will separate. One sister chromatid will go to one side of the cell. That's one green 3 going to one side of the cell. The other sister chromatid will go to the other side of the cell. That's the other green 3 going to the other side. That's exactly what we wanted to happen. We wanted identical chromosomes to go to opposite sides of the mother cell, and that's what we're going to get. But not yet. Before that, we need to move on to metaphase. Before the sister chromatids separate, so they're still held together by cohesin, all of this pulling and tugging, all of this tug of war, results in each of the 46 chromosomes being neatly and orderly aligned at the very center of the cell. And they are aligned, what we say, head to toe or head to foot. They are single file, as shown in this diagram, single file across the entire center of the cell. We call this cell center, this line which the chromosomes are lined up on, the metaphasic plate, or the metaphase plate. Remind you, the centrosomes are on opposite sides of the cell still. They're sending out their spindle microtubules. The microtubules are attached to each of the kinetochores on each of the two sister chromatids, but the sister chromatids are still held together by cohesin. And so we have an equal and opposing force being exerted on these chromosomes, keeping them lined up right in the middle of the metaphasic plate. Now this alignment is checked by the cell. This is actually a mitotic checkpoint. The cell checks this lining up by simply measuring that there is equal tension on each of the sister chromatids, making sure that there truly is a tug of war. It's amazing. Think about all that's going on here. This is happening inside of you right now. Wherever you're sitting, wherever you're listening to this lecture, some of your cells are actively dividing. Your skin cells, the cells of your digestive system, those cells are actively dividing. So right now, all of this complexity is occurring in some of your cells. Think about if this checkpoint wasn't occurring. Think about if the cell didn't care about equal tension on the line. What would happen if a single spindle, a single microtubule from one side of the cell was attached to one sister chromatid, and the spindle on the other side of the cell, the microtubule from the other centrosome, was not attached to the other sister chromatid? So let's imagine that. What I'm saying is that one centrosome has sent a microtubule successfully to this kinetochore, attaching to this sister chromatid, but the other microtubule from the other centrosome missed, and so nothing is attached to this sister chromatid. Now they're going to try to play tug of war, and when they do, I said it the opposite way, but when they do, the centrosome that sent out the correct attaching microtubule is going to win that tug of war game right away. It's going to pull the entire chromosome to one side of the cell. When that happens, eventually, the cell on the right is going to have two chromosomes, the two sister chromatids we see here, and the cell on the left, which didn't correctly attach, is going to have no chromosomes. Now that's a problem. That's a problem because too many chromosomes is problematic for a cell. We know that. That's actually what causes Down syndrome when this occurs during meiosis. Too few chromosomes is also a problem. There's genetic information on that sister chromatid, and the daughter cell needs that genetic information. So if the daughter cell doesn't get it, it doesn't have the full complement of information it needs. So if there's not equal tension in that tug-of-war game, if both sides of the cell, if both centrosomes are not truly vying for this chromosome, if there's not equal tension across these two, then the cell knows that the sister chromatids are not attached on both sides. And so these cells are blocked for continuing on with mitosis. It's better to stop here in the middle of mitosis than continue through with cell division and have two new daughter cells made that each contain the wrong number of chromosomes. If mitosis wasn't blocked, we would have two new cells with the wrong number of chromosomes, and those cells would be defective. So let's assume that there is equal tension across all 46 chromosomes, all of our microtubules have attached properly to all of our kinetochores, and we pass that checkpoint. Now we move on to anaphase. With equal tension across our spindle, we move on to anaphase. Now the glue that was holding the sister chromatids together, called what? Called cohesin, that's right, breaks down, allowing the sister chromatids for the first time to separate. So now we will begin to pull identical sister chromatids to opposite sides of the cell. Because of all that pulling and tugging of the microtubules, as soon as cohesin is dissolved and the sister chromatids are released, they are pulled to opposite sides of the cell. This means that we now truly have two identical sets of identical chromosomes moving in opposite directions to opposite sides of the cell. What do you think happens next? Well, if you said the cells are going to begin to divide, you'd be right. And that happens in telophase. Don't say chromatids anymore. Don't say sister chromatids anymore. Don't say chromatids anymore. Don't say sister chromatids anymore. Why not? Well, the term sister chromatid refers to two identical chromosomes that are physically stuck together. Once we proceed past anaphase, we separated those molecules into two independent molecules. They're not stuck together anymore. So we no longer have sister chromatids, we have independent chromosomes. Once those independent chromosomes arrive at opposite sides of the cell, called the spindle poles, we've entered telophase, the last phase of mitosis. A nuclear membrane forms around each new set of chromosomes, resulting in two new, distinct but identical nuclei, each containing an identical set of chromosomes. The chromosomes decompact. They lose that denseness that they took on at the beginning of prophase. They no longer are visible in the light microscope. They are now accessible and can be used as instructions by the cell. And in many cells, cytokinesis also occurs simultaneously with telophase. Cytokinesis, again, is true cell splitting. I think Table 2.1 in your textbook is extremely beneficial. I could see this being a very valuable study guide. This is that table reproduced here for you to take a quick look. It gives you a very nice rundown and a very brief synopsis of what occurs in each of the stages of mitosis, its name, and the main cellular process that occurs there. Very, very distinct, succinct breakdown of what I've just told you about. I'll give you the mnemonic now because I think it makes some sense to, but I'll have it on the slide later on as well. The mnemonic to remember the order starts with interphase and brings you through the rest of the phases. Interphase, prophase, prometaphase, metaphase, anaphase, telophase. And the mnemonic is, I proudly prefer milk and tea. If you can remember that phrase, I proudly prefer milk and tea, then you should be able to remember the order of interphase, prophase, prometaphase, metaphase, anaphase, telophase. It should go without saying, but let's say it anyway. Once mitosis is complete and cytokinesis is done, each new daughter cell should have a genome that is identical to the mother's genome before cell division. That was the point of mitosis. No DNA lost, all DNA accounted for, every cell gets one and only one copy of the chromosomes it needs. Now, although the genomes are identical, the organelles and cytoplasm is not. The cell goes through no great pains to make sure that the cytoplasm and organelles are distinctly and exactly separated in two. It's sufficient that some of the cytoplasm goes to one daughter cell and some to the other. Some of the organelles go to one and some go to the other. So the cell doesn't go through great pains to do that. There's no way to do that equally, and the cell doesn't bother to. It's just important that each daughter cell gets some. This is the full figure from your textbook that goes through each of the panels that I kind of broke down for you as we went along. I won't belabor it, but interphase is where we have all the other cellular processes of the cell cycle occurring. Prophase begins with DNA compaction and the centrosomes moving to opposite sides of the cell. They begin forming their spindle microtubules, but they can't yet hook onto chromosomes because the nuclear envelope is still there. Prometaphase is marked with the disintegration of the nuclear envelope. As we go into metaphase, the microtubules from the centrosomes hook on to the kinetochores of the chromosomes and align them in single file along the center of the cell, the metaphasic plate. Anaphase is marked by the breakdown of cohesin, allowing the two sister chromatids to become true, independent, distinct chromosomes, and they're pulled to opposite sides of the cell. We then move on to telophase, where two new nuclei form around those two sets of chromosomes that have been pulled to opposite sides of the cell, and cytokinesis or cell splitting can occur. Here's some questions for you to ponder. I'm not going to give you the answers to these questions here. We can talk about them in class on Monday if you'd like, but just some things to think about and make sure that you're understanding these concepts correctly. So, a human skin cell has 46 chromosomes, as all human cells do, 23 from mom and 23 from dad. How many centromeres does that skin cell have? Now take the same cell, imagine that it is undergoing mitosis, and it is in metaphase of mitosis. How many chromosomes does it have now? How many centromeres does it have now? How many sister chromatids does it have now? How many kinetochores does it have now? Take that same cell, proceed on to the middle of anaphase. How many chromosomes does that cell now contain? How many centromeres? This figure should help you get to those answers, at least logistically. You can pause the lecture here if you'd like and take a good look at this figure. But if you can't get confident, correct answers to these questions, you do not have a thorough understanding of what we've described in mitosis. If you can get correct answers to these questions, you probably have a pretty good understanding of what's going on in the stages of mitosis. Just something to keep in mind. All right, so now we're going to proceed on to cellular reproduction. That is sexual reproduction, and talk about why that's important, what benefits sexual reproduction gives us, what we need for sexual reproduction, and I've already kind of given that away in the beginning of this lecture, but that's going to lead us straight into meiosis, and we'll end the lecture with that. So we're well past halfway in this lecture, but we've got quite a bit of material left. So if you do want to break this lecture up into two, if you think you've gotten enough and you're not really starting to think clearly anymore and all that mitosis stuff really freaking you out, I suggest that you stop the lecture here, kind of decompress, pretend this is the end of a single lecture session, and then come back to this lecture at a later time as we proceed into meiosis. So this is a good breaking point if you'd like to kind of split this lecture into two. So if you did take a break, then welcome back, and if you just kept on trucking through, then kudos to you, I guess. But the good news as we proceed on to meiosis is that much of what's in meiosis is very similar to mitosis. So if you've become somewhat comfortable with the mitosis material and you think you have a good handle on those stages and what happens in them, we'll be able to recycle and reuse that knowledge for meiosis, and you'll see that in just a second. But before we talk about meiosis itself, let's have a brief discussion on why it's even needed. Meiosis is a specific cellular process that allows sexual reproduction to occur. Meiosis makes gametes, and gametes fuse to create new life. Now, there's plenty of evidence out there, as I'm sure you know from general biology, that asexual reproduction works fine too. Many organisms reproduce asexually. One organism splits into two. Bacterial cells do that, but we see examples of that all the way up through the kingdoms of life. So, What's the benefit of sex? Why go through all this extra trouble to create these gametes and create new novel life when it's much easier and more straightforward to make life from a single individual? So I just explained to you in the mitosis information, and hopefully I taught you in a way that you could understand how a single cell can become two identical cells, identical genetically speaking at least, and that's asexual reproduction. The point of life is to, in essence, divide, reproduce, continue to survive, and make it to the next day. So if you can do that asexually with one individual, why have a sexual cycle? Well, the real answer boils down to things change. Things change. Let's say the world was real hot. Really, 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 really hot. And all the cells on that planet, on this planet, were adapted to that very hot environment and had genes in their DNA encoding proteins which do everything, remember, that allowed them to survive on that really hot world. Hey man, that's great. You're going to have these single individuals perfectly adapted to this hot planet that continue to divide and thrive. But things change. That world begins to cool, and it cools down pretty quick. And now the cells that exist on that planet to survive need to be able to have genes, to have genetic information to build proteins that will allow survival on a cooler world. With just mitosis, with just asexual reproduction, with one individual becoming two identical individuals, too friggin' bad. Your genes don't cut it. You're not going to make it on that cool world, and your species will become extinct. Mitosis gives rise to genetically identical daughter cells, cells that are adapted for the here and now, sure, but cells with no potential to change for the environment of the future. To survive on a changing world, to survive on a dynamic world, we need variability. We need rapid changes. We need differences among individuals. We need sex. And that's where meiosis comes in. Evolution. Survival of the fittest, given that current environment that the species is currently in, depends upon genetic variability, depends on differences, depends on a species that has individuals that are different, not identical. Survival of the fittest, even in that one term, implies that some members of a population will be better for a given environment than others. If some of us are more fit than others, then we are all different. And if we are all different, we require variability. And if that's the case, asexual reproduction just ain't going to cut it. We have to be different. Sex shuffles genetic information of two parents and creates a completely new, novel, and unique genome different from each of those parents, giving a different individual a brand new human being who is different. These are my two new human beings that are certainly different. This picture's a little bit old. You've seen these guys in the course trailer already. Charlotte's on the left and Madeline's on the right, Charlie and Maddie. They are not me. They are not my wife, but they are similar to each of us in very distinct and recognizable ways. They are also different from each other, although they have the same mother and father, because sexual reproduction has reshuffled my genetic information and that of my wife's, creating a unique and novel human being who has never been on this planet before and will never be on this planet again. When we have this variability, when we have these individuals that are different, we have people that are more fit than some and less fit than others, and that drives evolution. So to create this variable population, to create billions of people who are all different, we require a mechanism by which a father can contribute half of his genome to an offspring, a mother can contribute half of hers, and these two half genomes come together to create a cell with exactly one entire and novel human genome. Meiosis makes that possible. Meiosis is the cellular process that creates a cell with a half genome, solely for sexual reproduction. Now, logistically, from the student perspective, the good news about meiosis is that much of it is identical to mitosis. Both of these stages consists of cells that go through the cell cycle. So both meiosis and mitosis have cells that are coming out of interphase. Meiosis includes two stages with cellular divisions. You'll see why in just a second. We refer to them as meiosis 1 and meiosis 2. Meiosis 1 is referred to as a reductive division. Something is being reduced. Maybe you can guess what it is. When cells divide after meiosis I, they do so without separating their chromatids. This results in the number of chromosomes being halved. That's what we want. A cell with half the number of chromosomes. Meiosis II looks virtually identical to mitosis. You can have no problem with that at all. Sister chromatids separate. Those sister chromatids are pulled to opposite sides of cells. Cells divide. Half genomes are maintained because we achieve the half genome in meiosis I, and you've got yourself a finished product. Let me explain it this way. I think hopefully it'll make a little bit more sense. Here is our initial cell. Okay, this is a diploid cell. I have one large red chromosome from my father and one large blue chromosome from my mom. They're a homologous pair. They're similar but not identical. I have one smaller red chromosome from my father, one smaller blue chromosome from my mother. This is also a homologous pair. Now this cell has already undergone interphase. So DNA replication has occurred. So actually these 4 chromosomes are actually 8 sister chromatids. We should be comfortable with that by now. But there are 4 chromosomes in this cell as we see it. DNA replication has occurred. We have 8 sister chromatids. These 2 chromatids are identical copies of my father's. These 2 are identical copies of my mother's, and so on. There are 4 chromosomes here. After meiosis I, each new cell after the cell division gets only one of the chromosome number 1 chromosomes, and only one of the chromosome number 2 chromosomes. The sister chromatids are still held together, but only one of the homologous members, dads or moms, goes to the new cell. So how many chromosomes are in this cell? Hopefully you said 2. 4 sister chromatids, but I asked you about chromosomes. 2 chromosomes in this cell up here, and 2 chromosomes down here. Take a step back. How many chromosomes were in the initial cell? 4. How many in the new cells? 2 and 2. That's your reductive division. The number of chromosomes has been reduced by half. Now in meiosis II, we're going to have sister chromatids separate. So these sister chromatids separate. One new cell gets one of them, and the other new cell gets the other. These 2 sister chromatids in blue separate. One cell gets one, the other cell gets the other. How many chromosomes in this cell that I'm indicating here? 2. How many chromosomes in this cell? 2, because the sister chromatids separated, and 2 here. So this is not a reductive division. This is not a reductive division. 2 chromosomes were split into 2 chromosomes because sister chromatids separated, and that's what occurs in meiosis II. So we go from 46 chromosomes in a human cell. After the first reductive division, we're down to 23. But then sister chromatids separate, so we get 23 after the second meiotic division as well. And that's what we want, cells with 23 chromosomes. Meiosis II is sometimes called an equiational division, and you can see that labeled here. I think that's a stupid word. I'll probably never use it again, but reductive division certainly captures what's occurring in meiosis I. So let's go through meiosis, stage by stage. We'll kind of skirt over the details that are similar to mitosis and highlight the differences. Prophase I, this is prophase in meiosis I, is a very complicated and involved affair. A lot happens in prophase I, and we'll spend most of our meiosis time talking about this one stage. Because it's so complicated, prophase I is actually divided into 5 substages. They're poorly named. They're given complicated names that I will never tell you, never use, and never hold you accountable for. You'll see them in just a second because they're on a figure. But don't worry about the substages of prophase I. Just know what happens in prophase I. To keep things somewhat simple, I'll not break prophase I down into these substages. If it helps you to break things down and compartmentalize, feel free to use the text to kind of do that breaking down with those names. It's up to you. The first thing that happens in prophase I that is of any consequence is the same thing that happens in prophase of mitosis. The chromosomes compact, they become very dense, and they become visible on a light microscope. Next, in prophase, homologous chromosomes line up next to each other and touch. Now let me emphasize this. Homologous chromosomes, not identical sister chromatids, homologous chromosomes line up next to each other. In mitosis, homologous chromosomes lined up head to foot, single file. Not so in meiosis. Okay? These are the homologous chromosomes, one from mom and one from dad. It's this close association of homologous chromosomes that occurs only during meiosis, because in mitosis they're single file, and this close association is referred to as synapsis. Synapsis only occurs in meiosis 1. During synapsis, when these homologous chromosomes are touching next to each other, we have 4 chromatids that are very, very similar, if not identical, closely associated with one another, and we call this structure a tetrad. Tetra for 4. Get it? Tetrad. A tetrad is 4 chromatids side by side. It is in synapsis where the genome shuffling truly occurs, and this is referred to as crossing over. Crossing over not like dying, not crossing over to the other side, but the crossing over of genetic information. So this is prophase 1 in all of its complicated glory, and these are the names that I referred to earlier of the substages, but I'll not use those. The end result of prophase 1 is synapsis, as shown here. Homologous chromosomes side by side. Your 4 chromatids, 1, 2, 3, 4, all side by side, creating your tetrad. These chromosomes are in synapsis. So what is crossing over if it's not dying? Well, crossing over is when chunks, large stretches, large regions of homologous chromosomes, not identical, homologous chromosomes are traded between each other. This creates chromosomes in sex cells, because this is unique to meiosis, that never truly existed in the parent. Think about that. The level of variability, the reason why parents can have 14 kids and none of them are identical to one another unless they're identical twins, is because of this genome shuffling. I have passed chromosomes on to my children that I myself don't have because of crossing over. This means that after prophase 1, it's quite possible that none of the 4 chromatids in the tetrad are identical. So let's look at this in pictures to kind of make it have a little bit more sense. Here we have the beginnings of crossing over. I have my paternal chromosome in light orange. It has already been replicated, so I have 2 sister chromatids here, okay? And my maternal chromosome in dark orange, already been replicated, 2 sister chromatids, and they are in synapsis. They are lined up side by side. They are creating a tetrad, 1, 2, 3, 4 chromatids, and this is in prophase 1 of meiosis, okay? Now, crossing over means we're trading chunks of genetic information between the touching chromatids. So when synapsis or crossing over is done, we see that this segment of my mother's chromosome, which she gave me, is now popped onto this chromatid from my dad. And this piece of genetic material had initially come from my father, but it's popped onto this region of my mother's genetic information. In this case, when these sister chromatids separate, we're going to have one that is all orange, one that's mostly orange with a little from mom, one that's mostly mom with a little from dad, and one that's all dark orange from mom. 4 different chromatids. 4 different chromatids. In addition, the whole idea of having genetic information passed on that never truly existed in the parent. See, in all of my cells, I have 1 whole chromosome from my dad and 1 whole chromosome from my mom. That's what makes me me. But if I give this, let me go back to my little drawing tool. If I give this chromosome, once it's separated, this chromatid to my daughter, I'm passing on genetic information, I'm passing on a chromosome that is largely my dad's with some of my mom's information on it. I don't have any single chromosomes in my cells that are mostly dad and some mom. Half of my chromosomes are all from my father, and half of my chromosomes are all from my mother. But my kids can get a shuffled version of that, which never existed in myself. That's the result of crossing over, and it adds an enormous amount of genetic variability to sexual reproduction. Crossing over is the basis of recombination, recombining chromosomes into new combinations. We also refer to this as genome shuffling for obvious reasons. Crossing over provides much of the genetic diversity we get from sexual reproduction because we take existing genomes, like the one that I have in me, and recombine them in new configurations that are novel to any child that comes after me. But crossing over can't occur in mitosis. Why? Think about that. Why can't crossing over occur in mitosis? This image on the slide right now is a dead giveaway hint. Homologous chromosomes never occur side by side in mitosis. They only line up single file. What allowed crossing over to occur was synapsis. What allowed synapsis to occur was the lining up of homologous chromosomes side by side. Without side by side alignment, there is never synapsis, and without synapsis, there is never crossing over. So in mitosis, we don't have synapsis because we have single file alignment of homologous chromosomes. In meiosis, we do have synapsis because homologous chromosomes line up side by side. This allows us to pass DNA molecules on to our children that we ourselves never had, enormously increasing the genetic variability in our species. So after some of this crossing over, we see here's the parental cell, one chromosome from dad, one chromosome from mom, dad in orange, mom in blue. We have DNA synthesis and interphase, so now we have sister chromatids. In prophase 1 of meiosis, we have crossing over due to synapsis, and now after meiosis 2, we have 4 new cells. One cell has dad's entire chromosome. One cell has mom's entire chromosome. One has one that is largely dad's with a piece of mom's, and one has one that's largely mom's with a piece of dad's. After all that crossing over, prophase 1 ends with the disintegration of the nuclear membrane, the formation of mitotic spindles, all of that very, very similar to what we saw in mitosis. Metaphase 1, that is metaphase of meiosis 1, has homologous chromosomes. Again, homologous chromosomes align at the metaphasic plate. Microtubules attach to each of the chromosomes, not each of the sister chromatids, each of the chromosomes. Pay attention to the figures as well. And anaphase 1 draws those chromosomes to opposite sides of the cell. This is our reductive division. This cell, as we see it now, contains 4 independent chromosomes. 8 chromatids, fine, but 4 chromosomes. In telophase 1, new nuclei form, cellular division is allowed to occur, and now that single cell that had 4 chromosomes in it is going to give rise to 2 new cells, each with 2 chromosomes. Each with 2 chromosomes. This cell has 2 chromosomes, 1, 2, and the other cell after telophase 1 will have 2 chromosomes, 1, 2. That is your reductive division. That is changing 4 chromosomes into 2. Cells take a brief break. They essentially reset. Actually, the DNA decompacts and then begins to recompact. Cells then enter meiosis 2. Meiosis 2 is almost identical to mitosis. Very little to discuss here. Prophase 2 begins with chromosomes recondensing. They become visible under a light microscope yet again. They had relaxed during the break, as I just mentioned. These chromosomes consist of 2 attached sister chromatids being held together by cohesin. They're not necessarily identical. Certainly after crossing over, that's the case. But they are still sister chromatids. Spindles form in prophase 2, the nuclear membrane begins to break down in prophase 2, and then cells enter metaphase 2. Metaphase 2, just like metaphase of mitosis, has us see chromosomes line up in single file at the metaphasic plate, as we see up here. They're just turned on their side. In anaphase 2, those sister chromatids are pulled apart. Cohesin has dissolved. Sister chromatids are pulled apart. They're pulled to opposite sides of the cell as those microtubules play tug of wars. All of this occurring due to microtubules attached to kinetic cores of sister chromatids. Do you see how many chromosomes are in this cell? 1, 2, 3, 4. Can you anticipate how many chromosomes are going to be in each of the daughter cells? 1, 2, 3, 4. 1, 2, 3, 4. This is not a reductive division. Because sister chromatids are separating, the chromosome number stays the same. Finally, in telophase 2, 2 new nuclei form. the cells divide as the DNA relaxes, and we have our end result of meiosis. So what just happened? Well, another good study guide, Table 2.2 from your textbook, breaks down each of the phases of meiosis, both meiosis 1 and meiosis 2, and tells you what occurs in each of those phases. Prophase 2 refers to the synapsis, and crossing over, reminding you that occurs in prophase 2. I highly recommend you make use of this study guide as a resource. To boil it down, though, in meiosis, we took one cell, which had 46 chromosomes if it was human, and made 4 cells, each with 23 chromosomes. We took one cell with 46 chromosomes, divided it into 2 cells with 23, and then separated sister chromatids to make 4 cells with 23 chromosomes, with reshuffling, crossing over genetic recombination, adding variability. We were able to do this with 1 replication of our DNA and 2 rounds of cell division. Now these 2 cells with 23 chromosomes each can go off and fuse with another gamete with 23 chromosomes, making a new human being reconstituted with 46 chromosomes total. Dad's sperm has half the number of chromosomes needed to be a human being. It fuses with mom's egg, which has half the chromosomes needed to be a human being, creating a full complement of genetic information and therefore a new human being. As part of this, we had a hell of a lot of genetic diversity introduced, but there's more. Not just crossing over is responsible for the differences between siblings. We have something called random separation, or more commonly referred to as independent assortment, that gives us genetic variability as well. I emphasize independent assortment now because independent assortment is going to play a huge role in the more complicated stages of Mendelian genetics that we'll touch on in a few weeks. Crossing over adds tons of genetic diversity to sexual reproduction. There's no doubt about that. But it's not the only thing doing so. The other phenomenon adding to genetic diversity is independent assortment. Said briefly, independent assortment refers to the random distribution or separation of homologous chromosomes during meiosis. The textbook, I think, is great. Largely, it's well written and clear. But the textbook makes independent assortment slightly unnecessarily complicated, when to me, it's really a pretty easy concept to get your mind around. Here's my genome. And as my daughters would say, not for real, just for fake. This isn't really my genome, but it's an XY genome, so it will do. In my genome, I have 2 homologous chromosomes for each homologous pair. I have 1 chromosome 1 from my dad and 1 chromosome 1 from my mom. 1 chromosome 2 from dad and 1 chromosome 2 from mom. We've been through this a number of times. Let's assume, for the sake of argument, that every chromosome you see on the left of the pair came from my father, and every chromosome you see on the right of the pair came from my mother. And as you can see, I got ahead of myself, so let's scratch all that and stick to what the notes say, make it easier for everyone, and say that mom is on the left and dad is on the right. Okay. And that's for every single chromosome pair that you see. Independent assortment, this idea of independent assortment is quite simple. It states that there are no rules as to which of these chromosomes a given gamete gets. In other words, it's a mix and match on what gamete is going to get which chromosome. It's important that every gamete get 1 and only 1 chromosome number 1, 1 and only 1 chromosome number 2, 1 and only 1 chromosome number 3, but that's the only rule. That's the only rule. So there's a gamete out there of mine that has only my mother's chromosomes in it. That gamete got my mom's chromosome number 1, my mom's chromosome number 2, my mom's chromosome number 3, my mom's chromosome number 4, my mom's chromosome number 5, blah, 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 blah. There's a gamete out there that got my dad's chromosome number 1, my dad's chromosome number 2, my dad's chromosome number 3, my dad's chromosome number 4, blah, 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 blah. That's perfectly fine. Doesn't break any rules. As long as that gamete gets 1 and only 1 of each chromosome, it satisfies the rules of meiosis. But independent assortment says you can do whatever the hell you want. There can be a gamete that has mom's chromosome number 1, mom's chromosome number 2, dad's chromosome number 3, mom's chromosome number 4, dad's chromosome number 5, dad's chromosome number 6, mom's chromosome number 7, blah, 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 blah. That gamete is going to have one and only one of every chromosome pair, satisfies that rule, and that's the only rule. But independent assortment says every single possible combination is possible. Dad's 1, dad's 2, mom's 3, mom's 4, mom's 5, mom's 6, dad's 7, dad's 8, mom's 9, fine. Another combination that goes every other. Mom's dads, mom's dads, mom's dads, mom. All of it is possible. It winds up being 8,388,608 different possible combination of gametes you can make by just independently assorting mom's and dad's chromosomes. And listen to this. That doesn't take recombination crossing over into account at all. That's 8,388,608 different possible combinations without any recombination or crossing over. It's astounding. So the textbook says, crossing over shuffles alleles on the same chromosome into new combinations. It creates new chromosome molecules with new combinations of alleles. Whereas independent assortment zooms out one level and shuffles alleles on different chromosomes into new combinations by taking different sets of mom's and dad's chromosomes and mixing them together into a single gamete. Together, these two processes, crossing over and independent assortment, are capable of producing mind-bogglingly high amounts of genetic variation among cells resulting from meiosis. This is why, no matter how many children a mother and father have, it is statistically impossible that anything but identical twins be identical. The variability is just far, far too great, and that was the whole friggin' point from the beginning, the variability. Making different individuals in the population with different fitnesses so that anybody can survive and thrive as the most fittest individual in a given environment. This table from your textbook, Table 2.3, is a very useful comparison between the three phenomena we just discussed, mitosis, meiosis 1, and meiosis 2. Is there cell division in mitosis? Yes. Is there cell division in meiosis 1? Yes. Is there cell division in meiosis 2? Yes. Are chromosomes reduced in mitosis? No. The chromosome numbers kept the same. It's not reduced in meiosis 2 either. That's an equal division. But there is a reductive division in meiosis 1, so yes, chromosome numbers reduced. Do you get any genetic variation in mitosis? None at all. Do you get it in meiosis? Oh yeah, that's where your recombination occurs. But meiosis 2 doesn't introduce any additional genetic variation. Where does crossing over occur? Well, you can go through this chart, but I find this chart to be quite useful. Okay, so let's wrap up and talk about meiosis in animals such as us and end the lecture with that. The production of gametes in any male mammal, any male animal, occurs in the testes and is referred to as spermatogenesis, the making of sperm. We start with a cell that's referred to as a primary spermatocyte. This is a diploid cell that contains 46 chromosomes in humans, and it has entered prophase 1, so that marks it, makes it destined for meiosis. It's in prophase 1, we have synapsis, we have tetrads, we have the potential for crossing over, etc. After meiosis 1 is complete, we have a reductive division. We now have secondary spermatocytes with half the number of chromosomes as was in the primary spermatocyte. The product of meiosis 2 are spermatids, which have the right configuration and number of chromosomes, but are simply immature haploid sperm cells. They don't have a tail, they're not yet motile. These spermatids do mature into motile, free-swimming, fertilization-competent sperm, and they're done. That's meiosis in men. And it's pretty boring. There's not much to it. Men are boring in general. I think we all knew that. The production of gametes in female mammals and female animals is much more interesting. It's called oogenesis or oogenesis, depending on who you hear the word from. It begins much like it does in males with a cell that we refer to as a primary oocyte. The primary oocyte is diploid, as is the case in males. However, after the cell division of meiosis 1, we not only have a reductive division, we have an unequal division, meaning one cell is considerably larger than the other, and this is because the larger cell got most of the cytoplasm. The smaller cell got very little of the cytoplasm. The cytoplasmic division in men was equal. The cytoplasmic division in females is unequal, giving one very large cell, referred to as the secondary oocyte, and a much, much smaller cell, which really is nothing more than a nucleus, referred to as the first polar body. Genetically, DNA-wise, the cells are identical. They have the same DNA. But the first polar body has very little cytoplasm, very few organelles, and it's not viable. It'll die. The secondary oocyte then undergoes meiosis too. Here the chromosome number is kept the same, but we have another unequal cell division. One cell gets the vast majority of the cytoplasm, and the other cell gets very, very little. The larger cell this time is called an ovum. It is a mature, fertilization-competent cell. The smaller cell is referred to as the second polar body. Polar bodies, again, are not viable. They usually die off and disintegrate. Only an ovum can be fertilized. So where with each round of meiosis, male make 4 gametes from a single diploid progenitor cell, females only make 1, because the polar bodies die off as a consequence of the unequal cell division. So females start with 1 primary oocyte and end with 1 ovum. Males start with 1 primary spermatocyte and end with 4 spermatids. This figure from your textbook kind of shows this process quite well, I think, in my opinion. You can see that we actually start with a progenitor cell before even, that's referred to as a spermatogeum and an oogeum. I'm not going to hit you with those. I think those are overly complicated. So for our purposes, we'll start with the primary spermatocyte, the primary oocyte, both diploid. You can see here the unequal division. Here's the first polar body, it's much smaller. Here's the second polar body, it's also much smaller. Here's the single ovum that came from our single progenitor cell. Here are the 4 spermatids that mature into sperm. And of course, when sperm and egg fuse, we have a fertilized zygote that is diploid. That's our new human being. So take a moment and just reflect on why do you think that is? Why do you think that females only get 1 ovum for each progenitor cell and males get 4? The reason is the same reason it gives us a chicken egg. The female egg, even in the human being, provides the initial nourishment for the zygote. When that very small sperm cell fuses with the ovum, all that is delivered by the sperm is DNA. Really, that's all sperm is a DNA capsule with a tail. All of the mitochondria, all of the cytoplasm, all of the cellular machinery, everything that makes a cell, comes exclusively from the mother. The mother creates the developing cell that the new human being can develop from. Now, the ovum is not a human being because it contains only half the DNA a true human being needs. But as far as cellular components are concerned, the ovum is a fully functional human cell. All that's needed is another half genome to complete the DNA content, and that's what the sperm provides. So the ovum needs to be full size because the ovum is that first human cell. Mothers in all forms of life provide that type of nourishment, provide that type of sparked life, with males providing nothing more than half a genome. As usual, much true to our character, men being lazy and providing and doing as little as humanly possible, while females shouldering the burden of creating new life. So this was, like I said, quite a long one. We covered quite a bit. Of course, send me your reflections. Let me know what wasn't clear, what you'd like me to go over again in class. We will master this material given this lecture, as well as our review on Monday, and using this material a little bit more in depth on Wednesday. You will get this, but it will require some review and some studying on your part as well, of course. But let's talk about, in summary, what we covered today. We started off talking just about the differences between prokaryotes and eukaryotes, and some of those differences involved compaction of DNA. Humans have chromatin, that's the idea of wrapping DNA around histone proteins, whereas bacteria have the supercoiling, the rubber band experiment that we did. We defined diploid and haploid cells. Diploid cells have 2 homologous versions of each chromosome, one from mom, one from dad. Haploid cells have only 1 of each chromosome. Something for you to think about. True or false? Organisms that have more chromosomes are more complex. Hopefully you said false. The organisms that we discussed in this lecture that had the highest number of chromosomes were the crayfish and the horsetail plant, both of which are far inferior to me. I don't know about you. Chromosomes and sister chromatids is this other kind of troublesome area. I went to great pains to try to make that clear. Two sister chromatids are identical, but if they are stuck together by cohesin, you are looking at a single chromosome molecule. Only when sister chromatids are separate do we refer to them as independent chromosomes. We went on to discuss the cell cycle. The cell cycle is the story of a living cell. We talked about G0, where the cell does its cellular duties. G1 is the preparation for cell replication. We have a checkpoint. S is the DNA replication. G2 is the preparation for mitosis, another checkpoint. M phase is mitosis and eventual cell splitting or cytokinesis. We went over mitosis in some detail. Mitosis is the name of the process where eukaryotic cells divvy up identical chromosomes, giving two new daughter cells two identical complements of genomes that was present in the one mother cell. I already told you the mnemonic. I proudly prefer milk and tea. I recommend you memorize that phrase at least, allowing you to remember interphase, prophase, prometaphase, metaphase, anaphase, and telophase. You are responsible for the names of these stages, the proper order in which they occur, and the major cellular events that occur in each of them. Mitosis gives rise to only genetically identical daughter cells. Mitosis is a cellular version of asexual reproduction. There's no variability there. To survive on a changing world, we need genetic variability. We need sex. Sex drives evolution by creating different individuals with different fitnesses. Meiosis is the name of the process that eukaryotic cells use to obtain cells containing half genomes, haploid cells. These cells are gametes, and they can come together. Two gametes can fuse, one from a mother and one from a father, creating a new genetically novel individual with a different fitness. We talked about meiosis 1 and meiosis 2. In meiosis 1, chromosomes split, reducing the chromosome number in half. That's our reductive division. In meiosis 2, chromatids split, keeping the overall chromosome number the same. In meiosis 1, we have our crossing over occur, as well as our independent assortment. Meiosis 2 really doesn't introduce any additional genetic variation. We talked about independent assortment and crossing over as the two major mechanisms for achieving genetic variability in gametes. Independent assortment is the idea that any new gamete can get any combination of mom's and dad's chromosomes as long as the gamete gets one and only one of each chromosome number. Crossing over is the exchanging of chunks of chromosomes of chromatids during synapsis in prophase 1. We ended today's lecture talking about how eggs are made in the human or mammalian animal. This is the process of oogenesis, and we talked about spermatogenesis as well. Whew! Well, that was a lot. Keep in mind, no other single lecture will be like this. This really is 2 lectures in one. The only reason I'm presenting it as a single lecture is because it is really a single story. This is the only lecture you're responsible for going over this week, and we will smooth out the rough edges on Monday. As always, thanks for watching, and I look forward to going over this with you guys in class.